Let's look at what our MHC1 would look like. So here's any nucleated cell in our body, and for argument's sake, let's say donut shape is our self-antigen, looks normal. So we've got a self-antigen with MHC1 nucleated cell. Great. If there's surveillance going around, it's going to look at that and go, oh yeah, that's a self-antigen, I'm good. Same here. MHC1 displaying a self-antigen, surveillance coming around going, oh yeah, that's normal, that's great. However, if MHC1 is displaying something foreign, notice these are not donuts anymore, right? This cell needs to die, this cell needs to die. It is still MHC1, but what I'm holding is different than what I should be holding. I should be holding something that is a self-antigen, and I am no longer doing that. Big red flag that there's something wrong. As far as MHC2, here's my antigen presenting cell with its MHC2 showing a foreign antigen on its surface. I'm not going to die because I'm not infected with anything. I just want to show you what I found. One of the, I guess, better ways that I can think to explain this, if I see a wanted poster for a serial killer, if there's a serial killer in town, and I see a wanted poster and I start showing it to people, should people kill me because I'm holding a wanted poster for a serial killer? I would hope not. I really wouldn't want to die because I'm trying to show you something that's important. That's what these guys are doing with MHC2. They're just showing you. They're not infected. Now, if I was the serial killer, different story. But I'm not. Same thing. Looking at what the interaction would look like. Here's my cytotoxic T cell with its CD8 molecule and its T cell receptor with its variable regions that recognize a specific foreign antigen. I've color-coded this so the variable region matches my foreign antigen. My T cell receptor binds to the antigen. My CD molecule, in this specific case, CD8, binds to the MHC. Having that bracket kind of bind allows my cytotoxic T cell enough time to kind of go through the steps of recognizing, I know what this is, I need to get activated and start making copies so that I can go after all of these infected cells. CD8 binds to MHC1. CD8 is only on cytotoxic T cells. Therefore, cytotoxic T cells are the only ones that even look at the MHC1 complexes. Now let's look at our helper T cell. Helper T cells have a CD4 bracket. CD4 only binds to MHC2. We'll never look at MHC1 at all. But again, I have two things binding, my T cell receptor and my CD4 bracket. The bracket binds to MHC2, my T cell receptor binds to the foreign antigen. When both are bound, it gives enough stability and time to get my helper T cell to go, okay, there is something in the body, I recognize it, I need to start making copies. So let's look at it in more detail. The antigen presenting cells, what are they? They can be macrophages, they can be B cells, and they can be dendritic cells. B cells technically are phagocytes, and I'll explain better when we get to the B cells. After the processing is complete inside of my antigen presenting cell, I'm going to blow this up so we can see it better. So here's my antigen presenting cell eating a microbe. I break it apart. We talked about this with phagocytosis. And I isolate the antigen, okay, the surface antigen. And I bind it to MHC2. Antigen presenting cells engulf a microbe, take it into the intracellular vesicle, degrade it into smaller peptides or pieces. The antigen pieces are complexed with MHC2. They're transported to the membrane and they insert into the membrane. Who's the only cell that recognizes MHC2? Helper T cells. So they're presented to the helper T cell. I need to go and show this to a helper T cell. The thing that makes it important, though, is I need to show it to the helper T cell that looks at this and goes, I know what that is. It recognizes that foreign antigen or the epitope. And FYI, it is possible, since these also have MHC1, since they have a nucleus, that it could be bound to MHC1, but in that case, it would be a death sentence. When I find the right T cell, helper T cell, I have my receptor bound to the antigen. I have my CD4 bound to my MHC2. The first MHC2 antigen on the antigen-presenting cell binds to the T cell receptor. Next, a co-receptor on the T cell, CD4, hooks itself into position on the MHC2. This two-step binding allows the helper T cell to acknowledge that it recognizes the antigen, and because it's being bound to MHC2, it's also the right MHC that it's binding to. Once that happens, the antigen-presenting cell activates the helper T cell by releasing interleukin 2. Once this is complete, once the binding has happened, it's a signal to my antigen-presenting cell to release a specific cytokine chemical that will transform this helper T cell into its active form. Now, the reason I'm being so vague on T cell and this chemical and all of that, we have at least 4 different helper T cells, and how they're activated also varies. I'm going to talk about that, but I want you to understand this is a very generic representation of how this works. Let's talk challenge and activation. Your book used the word challenge for my B and T cells. When challenged with a foreign antigen, when MHC1 shows up displaying a foreign antigen, whether it's 1 or 2, the T cells themselves, remember I said it releases interleukins, they will start to proliferate, they start to make copies, and they start to differentiate. A clone is a genetically identical cell. When I talk about that proliferation, I'm doing it by mitosis. I'm making an exact copy of what's already there. I make 2 populations. That's why I said differentiate. One population is going to be called memory cells. Memory cells go into hibernation and they wait. They don't do anything during this infection that we're dealing with right now. The second set of cells will be called effector cells. Effector cells have a job to do. Now, what that job is depends on what it's being made into, whether I'm a helper T cell or a cytotoxic T cell. It depends. So I'm just going to say for now, I make a population, usually a larger population, of effector cells and then memory cells that, again, get put away for later. The memory cells ensure that future reactiveness against that specific antigen, whatever it was presented with or challenged with. And as I said before, B cells and T cells, completely different jobs. I told you B cells are antibody factors, T cells are definitely not. There are 3 primary functional classes of T cells. We have helper T cells, regulatory T cells, and cytotoxic T cells. Each of these will secrete cytokines, which are chemicals, to help destroy pathogens, but again, not antibodies. Only one cell does that, the B cell, not T cells. Helper T cells have multiple jobs. We're going to talk about it, but just so you know, each helper T cell is activated under specific circumstances and then does something different. Regulatory T cells. These are kind of the guys that are going, dude, calm down. If your body has already taken care of an infection and it's under control and we don't need any more inflammatory chemicals and things like that, our regulatory T cells actually secrete chemicals that decrease the reaction that's happening. That's why we call them regulatory. They control the T cell response by secreting anti-inflammatory chemicals or cytokines, and they prevent proliferation. If the invader's already dying, I don't need bigger armies. my army is big enough. Cytotoxic T cells, cyto, cell, toxic, poison. These are what in A&P I refer to as the assassins. They actually release chemicals that kill the cells that have been taken over by an invader. Something that is important to remember with T cells, they only kill self cells that are infected. If there's a virus roaming around your body naked, these guys have nothing to do with it. Because I have to have MHC presenting me with something, it has to come from one of my cells. So they are effective against self cells that are wrong in some way, shape, or form. So for this example, I'm going to show you a helper T cell and kind of a really bad animation of what happens, but it's an animation of what happens. I'm not an animator, never claimed to be, but I like being able to see how it works. So here's my antigen presenting cell with its MHC2 displaying all of the foreign antigens that it has found. In this case, it was supposed to be a virus, just so you're aware. CD4 binds, my T cell receptor binds. Remember, this is kind of the key event to cause the chain reaction to help me to multiply this specific helper T cell, because this is the one that recognizes something off of my foreign invader, the epitope. Once it's stable, my antigen presenting cell releases interleukin 1. Interleukin 1 goes across to my helper T cell. This is going to cause a chain of events that leads to multiplication of the cell. Once that interleukin 1 has been released, that's a signal to this helper T cell to create interleukin 2 channels. So see these things? These are interleukin 2 channels on its surface. In order for it to start cloning, these need to be stimulated. Believe it or not, it's actually the helper T cell that creates the interleukin 2, releases it, and then it feeds back and binds to those receptors, causing the cloning to start. Remember what I said, I have two populations of cells. The memory cells get put away for later, and then I have the effector cells that do a job. Depending on which T cell you're talking about, the jobs vary. That's why I didn't put anything specific. The memory cells, though, they don't do anything right now. When challenged with the foreign antigens, T cells proliferate and differentiate. A clone or a group of genetically identical cells is created. Some clones are memory that will ensure future reactiveness. This is the same content as the previous slide. It's just after the animation. Let's get more specific. This has a lot of information. I have helper T cells type 1. I remember I said I have multiple helper T cells. Type 1, type 2, type 17, and then follicular helper T cells. You've got regulatory T cells, cytotoxic T cells, and gammadelta T cells. Gammadelta is that one that splits between innate immunity and specific immunity. They kind of work on both sides. T cell reactions are some of the most complex and diverse in the immune system, and they do involve, as you just saw, several subsets of T cells. The actions of the T cells are actually dictated by the antigen presenting cells that activate them. And you'll see why I say that in just a second. Their response is somewhat restricted because of the requirement for that MHC molecule. MHC has to have something on its surface so CD4 or CD8 will recognize it. That's why we say it's restricted, because this is a requirement. All T cells produce cytokines. Again, that's the general name for a set of chemicals. And depending on which cytokine it is, there is a spectrum of effects that happens. There are a bunch of different effects that can happen depending on which one you're releasing, which I will talk about. The end result of T cell stimulation, I am going to have mobilization of my T cells. I'm going to have mobilization of my B cells. And believe it or not, I'm going to induce my phagocytes to work even harder. Again, different cytokines, different results. So let's look at the T cell activation. Helper T cells. There are 5 types of helper T cells that all have that CD4 bracket that I've mentioned before. And they are critical. Remember I said everything hinges on. They're critical in regulating the immune reactions to antigens. They're also involved in activating macrophages, either directly via receptor contact or indirectly by releasing the chemical cytokines, such as interferon gamma. Some will secrete other chemicals that do other things. As far as the percentage of our lymphocytes that are made up of helper T cells, 65% of our T cell population, helper T cells make up 65% of our T cell population. So they're a big chunk of that pie. I very quickly went over this. Now let's go through the specifics. You guys see this. I just blew up the helper T cell portion of this. That's all I really did. We have the memory CD4 cells. If I am a T cell, a helper T cell that gets activated, I am going to make a colony of helper T cells that gets put away. So this isn't really a different kind, it's just the memory cells from these. I'm going to go down to the bottom here. This is the regulatory cell. This is the one that dampens my immune response, doesn't let it get out of hand. But I still have these 4. I have the follicular helper T cell type 1, helper T cell type 2, helper T cell type 17. All of these are activated for different reasons, and they produce different results. That's why they're separated. They're not just one type. A follicular helper T cell produces interleukin-4, and it produces growth factors specific to B cells. So when these chemicals are released, they activate the B cell, which turns into a regulatory cell, a plasma cell, and a memory cell. Just like our other memory cell, put away for later, it'll work in a second infection, it doesn't work now. The plasma cell is the antibody factory, so its job is to make antibodies. The regulatory cell does what the name suggests. It'll feed back and may suppress the T cell response if our infection has been dealt with. Helper T cell type 1. Now remember what I said earlier. They are activated for specific reasons, right? If we have an intracellular infection, we have viruses that have gotten into our cells and are mass producing viruses. This is the type of helper T cell I want. So my antigen presenting cell secretes interleukin 12 that will produce the type 1 helper T cell. What does it do? Well, it will release interferon gamma, and interferon gamma will stimulate my macrophages to become more bitey. They will start to eat more and usually work a little faster. They can also use these chemicals to promote antibody production that will in turn promote phagocytosis. I'm going to mention this here, but I'll talk about it more in a bit. If there's an antibody attached to something, phagocytes find it a lot faster and they eat it a lot better. It makes it more likely. So if I start the production of antibodies to attach to this invader's antigen, I'm actually helping my phagocytes out by giving them a flashing red light of where they need to go to eat. If we have a different infection. It's not intracellular. Specifically, if we have a parasitic infection. Parasites are things like pinworm and hookworm and tapeworm. These are very large things that invade our body. Phagocytosis is not an option in this case. And if I have antigen-presenting cells that come to realize this, they're going to go and find helper T cells and release a different interleukin. They're going to release interleukin 4. Interleukin 4 causes the creation of type 2 helper T cells. Type 2 helper T cells release 3 different chemicals that do 3 different things. 1, they release interleukin 4 that goes to B cells and causes them to make a specific type of antibody called an IgE, E as in egg, that coat the surface of the parasite. Another thing that this type 2 releases is IL-5, interleukin 5. Interleukin 5 activates 2 things, mast cells and eosinophils. If you think back to the last chapter, these are not phagocyte cells. They release chemicals. Both of these are going to be attracted to the IgE antibodies, and they are going to start releasing chemicals that are basically enzymes that will start to devour and break apart this worm parasite, the parasite that's there. The last thing that the helper T cell type 2 releases is interleukin 13. Interleukin 13 causes your digestive system to start mass releasing mucus, causing the inside of your intestine to become very, very slick. Why? It kind of increases the chances that these invaders in my intestine are going to get expelled. Since the sides are so slick, it's going to go. Type 2 helper T cells, completely different job than type 1. Finally, we have the type 17 helper T cell. For type 1, it was intracellular invaders, things that were inside of our own cells. Type 2, we had parasites. Type 17 is going to involve a fungal infection or a bacterial infection where they're not inside of our cells. The antigen presenting cell in this case is going to actually release interleukin 6 and transforming growth factor beta. Again, it's a different set of chemicals. It's going to cause a different result. In this case, that T cell is going to become a type 17. Type 17 releases 2 interleukin chemicals. Literally, interleukin 17, thus the name. Interleukin 17 is a chemical that attracts neutrophils to where this infection is. They also are a jumpstart for the inflammation process, and they defend against, the chemical itself defends against both fungi and extracellular bacteria that are outside of our cells. The other thing that it is going to release, that the helper T cell 17, type 17 is going to release, is interleukin 22. The lining cells, the endothelial cells, are actually going to themselves start producing antimicrobial peptides that are going to be on their surface, and those antimicrobial peptides resist infection. So we have different scenarios here. We have one scenario where our invader is inside of our cells. We have another scenario where we have a parasite like hookworm. We have a third scenario where we have fungi, a fungal or an extracellular infection. It's outside. It's not inside of our cells anymore. Either way, the T cells that are created are specific for those invaders. Different category are cytotoxic T cells. Cytotoxic T cells are in charge of something we call cell-mediated immunity. Mediate means to control. So cells are controlling this specific immunity. And the ones that do this are our cytotoxic T cells. Target cells that cytotoxic T cells destroy. What are their targets? Honestly, self cells that don't look right anymore. There are three primary ways that we have cells within our body that don't look like cells from in our body. First one is when we have an intracellular infection. So if a virus gets into our cell and starts reproducing parts of itself, and MHC1 grabs it and starts displaying this weird antigen, this foreign antigen on the surface, cytotoxic T cell is the one that recognizes that. So virally infected cells. Remember, cytotoxic means death. Cancer cells. Now I know you're going, wait a minute, that's not infected with anything. Cancer comes about because there has been some type of mutation to the genetic material. The DNA has been changed or altered in some way, shape, or form. Sometimes, not every time, but sometimes when that happens, you end up with proteins being created that aren't normal. If those are picked up by MHC1 and displayed on the surface, is a huge red flag to a cytotoxic T cell that there's something wrong with that cell. If it recognizes that there is something wrong with that cell because MHC1 is displaying something foreign, it'll kill it. For clarification, I know you're going, well, if that's the way it works, why do people still get cancer? I said sometimes when mutations happen, we get weird proteins. Sometimes when we get mutations, there is nothing weird that's displayed on the surface of our cell. So while they're doing surveillance and looking at the cells, they never see anything different. So they never attack it because it looks perfectly normal. That's the danger with cancer. The mutations may cause weird proteins to display, but sometimes it doesn't, because it doesn't happen in a place that has protein synthesis as part of its job. Cancer is more complex than just one scenario. However, if they are displaying a foreign protein on MHC1, cytotoxic T cells will normally deal with it and kill the cell. Finally, cells from other animals and humans, the most important factor in graft rejection. What do I mean? I'm sure by now you know about organ transplants. And I don't know if you know this, but when someone gets an organ transplant, they always do a tissue typing test. And one of the reasons is because they're trying to see what their surface antigen looks like compared to yours. Why would they do this? They want to make it as close to what yours looks like as possible, so that if they give you immunosuppressive therapy, these guys don't surveil and go, that's different, and kill that organ. It's something that does happen, even if they think they have a good match. But the initial setup is, I'm going to try and find something that looks as much like your surface antigen, your self antigen, as possible. The problem is, no matter what they put there, it is not yours. And with that in mind, depending on how different it is and whether or not the immunosuppressive therapy is actually effective, cytotoxic T cells can kill the implanted organ. The only exception to that is the cornea, at least that I'm aware of. And the only reason that works that way is because your cornea doesn't have a direct blood supply. You can donate anybody's cornea to anybody else, and since these cells never have access to it, it isn't normally rejected. Is there an exception? I'm sure there is. Not that I've heard of, but I'm sure there is, because this is science and there's always an exception. Gammadelta cells. T cell activation as far as cytotoxic T cells go. I do need to mention something with the cytotoxic T cells, and actually the B cells, it still applies. For these cells, it is a two-step activation. Being presented with the foreign antigen is not enough to activate this cell. When we talk about this in my other classes, I talk about starting your car. You put the key in the ignition. Is that enough to start your car? No. I can put the key in the ignition and sit there and nothing ever happens. The engine never turns on. Until you turn the key and it starts the engine, your engine doesn't turn on. So kind of a two-step thing. Put the key in place and then turn it to turn the car on. I know most newer cars have buttons, but just go with me on it. Cytotoxic T cells have the same type of two-step activation. The first step is what you're expecting. MHC1 and the foreign antigen are going to interact with my CD8 and with my T cell receptor. It will sit there forever if the second step doesn't happen. It doesn't do anything until helper T cell type 1 comes along, releases interleukin 2. Remember, my interleukin 2 had to get released to bind to the receptors to get this guy to start making clones? Same thing. Interleukin 2 gets released, and then this guy starts making copies. So step 1, binding to my MHC1 antigen complex with my T cell receptor and my CD8. Step 2, helper T cell releases interleukin 2. Then this cell, the CD8 cell, the cytotoxic T cell, starts to clone itself. I get memory cells that go off and hibernate till the next infection. But my activated cytotoxic T cell, also known as a CD8 cell, will recognize cells displaying the antigen that activated them. It releases two chemicals, perforins and granzymes. Perforins start to make holes in the membrane of the infected cell. If I make enough holes in you, you probably wouldn't like it either, right? Same thing. If enough perforin makes enough holes, it'll kill the cell. Granzymes are a bit more insidious though. As the holes are created, granzymes go in and they will invade the cell. Granzymes will cause the cell to go into apoptosis. Apoptosis is cell suicide. It's programmed cell suicide. So granzymes go to where this program is that causes this cell to commit suicide, and it turns that program on so that the cell kills itself. So you have perforins, granzymes. Either way, the cell that is housing that multiplying virus is going to die. Gammadelta T cells, they are distinct from other T cells. That's why we categorize them uniquely. They have T cell receptors that are arranged to recognize a wide range of antigens. So it's not just one, like my helper T cell and my cytotoxic T cell. They will frequently respond to certain kinds of PAMPs in the same way as our non-specific white blood cells, our macrophages and our neutrophils. Pretty much it responds in a very similar fashion, but they do respond faster than those cells, and unlike our non-specific, they do produce memory, which is weird because it's 1/2 and 1/2. That's why we always say that gamma delta cells are kind of splitting the fence between the two. They are particularly responsive to certain types of phospholipids, and they do react to tumor cells as well. And just like I said, they're kind of a bridge between non-specific and specific responses. The last two types of T cells I'm going to talk about are natural killer cells and natural killer T cells. Natural killer cells, these are actually categorized as part of our non-specific immunity, even though they're T cells. They don't have specificity. They do circulate through our spleen, our blood, and our lungs. And normally, they are the first killer cells that attack cancer cells and virally infected cells. They do use granzymes and perforins just like our cytotoxic T cell does, but they're not going to have the specificity and they don't create memory. They are sensitive to interleukin 12 and interferon though, which isn't necessarily true for our other non-specific cells. Natural killer T cells, they're hybrids. They're part killer cell and they're part T cell. They do have our T cell receptors for an antigen and the ability to release large amounts of cytokines very, very quickly that can lead to cell death. So they do have the ability to release granzymes and perforins a lot and very fast. But when you look at them, they're not the same as our cytotoxic T cells. They're not the same as our helper T cells. And because of that, we also consider them splitting the fence between non-specific and specific immunity. They're not necessarily one or the other.